Thank you very much, particularly for the pictures of your postdocs relaxing. Dr. Mitchel Doktycz has a BS in biology and chemistry and a PhD in chemistry from University of Illinois in Chicago. He is currently a distinguished staff scientist and leader of the Biological and Nanoscale Systems Group in the Biosciences Division of the Oak Ridge National Laboratory. Dr. Doktycz's research interests focus on the intersection of biological and nanoscale systems. Dr. Doktycz, could you come to the podium, please? So you have 20 minutes.
You just have to remember to keep looking this way. Well, I certainly want to thank the organizers for including me and giving me the opportunity to tell you about some of our work in trying to mimic cells. And I think like a lot of people at the conference, our application interests are very much focused on creating devices that can somehow sense and respond to their local environments. But beyond that, our interests are actually a little more general, a little more basic than that. We'd really like to start creating platforms, devices that allow us to ask very basic questions about the architecture and organization of cells and to test out systems. And our hopes and dreams are that if we could start mimicking cells, we really have a lot more applications open to us because cells are a very universal feature in biology and they do a lot more than just sense and actuate. They produce materials, they process information, et cetera. And so hopefully somewhere down the road we can have a very basic platform for doing a lot of different functions. And it really starts with understanding the cell. So there have been a few slides really highlighting sort of the scale of the systems we're talking about. And I really just want to maybe emphasize just a couple features here that I think have really spurred our interest in this field and kind of guides us in our thinking. One is, you know, with this nanoscience, we're getting this capability to engineer materials that are essentially on the same scale as the functional components of biological systems. And what's really critical about that is we need to interface to biology at its functioning level if we're going to understand it. And so my background has been in sort of technology development for genomics and post-genomics. And in this area, we develop tools to basically collect this parts list from biology. And so we have wonderful tools in genomics now to sequence genomes, look at proteomes, et cetera. So what results is this great parts list, but we really don't know how these parts come together to make systems and ultimately function in cells and higher order cells and communities, et cetera. This is obviously bacterial directed with communities. But if we could interface at these functioning levels, we have a chance to actually ask and answer how they work in real time. So the other point I want to make about scale is that if this was a physical science meeting, we'd really be talking about the wonders, the physical chemical properties that emerge at the nanoscale, much like you just heard with nanoshells, et cetera. And so there are wonderful enabling emerging phenomena that occur as a function of this nanoscale. And likewise in biology, function emerges because of the scale at which it's made and designed at. And these are things we would like to understand. And so comments about the importance of scale in biology. And one is the recognition that cells are basically a very conserved size. If you're a bacterial cell, you're typically just a few microns across. Eukaryotic cells are of course larger, but they tend to be compartmentalized and have different transport systems. And so if you just take a look at some of the statistics from an E. coli cell, it's a couple of microns across. There are about 50 million molecules packed in there. The components, of course, the proteins, et cetera, on the order of a few nanometers. And of course, that corresponds to volumes on the order of femtoliters. And so nature really isn't just showing off here. The point is that undoubtedly, this sort of architecture enables function. And one thing it enables is diffusion to be a very realistic transport mechanism. So it's estimated that any two molecules of a type will meet each other every second in a micrometer size volume. When you've got 50 million molecules packed into the size of a few microns, it means you get a very crowded environment. And so it's estimated that bacterial cells are on the order of about 20–30% crowded. And what that does is it changes your reaction rates, it changes your activity coefficients, it changes your diffusional flux across the system. Likewise, when you're at this sort of scale and volume, it means changes of just a few dozen molecules could really create potential gradients. And likewise, subtle changes in cell sizes change concentrations. And so all this basically enables biological function. And interestingly, this cell is the fundamental unit of all biology. And so if we could understand how the cell works, we could start understanding how higher order systems work and how these cells go together and how function emerges. And there are very interesting properties that basically relate metabolic rates, size, scale, et cetera, as well as longevity across the amazing diversities of biology. So the cell is a fundamental unit of biological systems. Its scale enables the processing of energy, information, and materials. That's a very engineering look at cells. So our interest is in mimicking cells, and over the last few years, maybe 5 to 10 years, there's been a variety of programs and phrases for this. Some people call them primitive cells, artificial cells, cell-like entities, et cetera. Likewise, we're starting to see the emergence of synthetic biology, and some people would call it metabolic engineering, et cetera. And our interests really are captured more by this phrase of nano-enabled synthetic biology. And what we'd like to do is to start transferring and understanding cellular attributes to synthetic systems and start merging sort of this bio and nano together. And I think this Feynman quote kind of captures a lot of this. If we could start creating these and understanding them from the bottom up, we're really going to understand how biological systems work. So, cell mimics. This basically is sort of an overview of the approach that we're taking. The idea here is just to simplify the cell into 2 basic parts here. One is this membrane. It's this membrane that really defines the volume, the concentration. It controls the input-output to the device. And of course, it's these molecular networks that we're trying to figure out where this input and this sensing comes about. You get signal amplification, you get logic, you'll generate output, you metabolize energy. And of course, all this goes on in parallel, and that's what's really wonderful about a cellular system. And so we think if we could emulate these parts, we'll have this universal platform. Now, what we're going to do here is we want to replace the membrane. We start here. We're going to replace the membrane with synthetic and nanofabricated structures. And this will then allow us to define self-sized and hopefully predictably control flux in and out of the system. Now, molecular networks are certainly still being figured out by genomics. And where we'd like to start here is by simply just taking DNA-based instructions, inserting them inside our synthetic containers, and then let machinery, cell-free extracts, convert these proteins, convert DNA essentially into proteins and hence start creating simple systems based on that. And of course, in this sort of structure, we could easily modulate gene copy number, codon usage, cross-reactivity, et cetera. And so this is where we could start understanding the effects of scale, flux, as well as transcription systems without poisoning the cell. And so we hope this will be a starting point for understanding biological systems and, of course, ultimately creating practical devices. So if you're an experimentalist, the cartoons are usually pretty easy to draw. It's how do you actually do this? And there are a lot of challenges intrinsic to this problem. First one is, of course, how do you make a synthetic membrane? We'd like to make fairly robust structures. We'd like to control their nanoscale features. We want a pattern-defined transport into them, et cetera. And of course, once you make that, how do you characterize them? And that's just not experimentally, but also theoretically. How do you actually observe what's going on a scale that's below the wavelength of light for the most part? Then just from a fabrication point of view, what's critical about biology is integrating structures across multiple length scales. And so how do you get nanoscale detail across multiple meters of an organism, let alone across a wafer scale? And then what further complicates this is we're trying to make systems that basically integrate soft materials, hard materials, and liquids while using vacuum-based devices to do that. And so a lot of this is aided by basically incredible resources at our national labs, as well as certainly universities have some of this as well. At Oak Ridge, we have the Center for Nanophase Materials Sciences. It's one of the 5 Department of Energy user centers. All of you are welcome to use it. It doesn't cost you any money other than your time and a 2- or 3-page proposal that gets approved by an external review committee. And so at this facility, it's 90,000 square feet of basically the latest, greatest tools for doing nanoscience, theory, calculation, imaging, et cetera. So our membranes are based on a couple of different nanostructures. These are carbon nanofibers. They're chaotically grown in a stripe here. And the way we're trying to make a membrane out of these is by controlling transport perpendicular to their orientation. And so we're taking advantage of the interfiber spacing to cause physical pores. We've got a lot of capabilities in this area. We can put individual fibers where we want. We could shape them, et cetera. We could integrate them into fluidic devices. It's kind of tough. This is probably a graduate student year's effort just to figure out how to do this. But basically, we want to take these platforms and design them. They're fairly robust. They're fairly stable. They withstand all the processing involved. And so as opposed to biological membranes, we can make these structures and they'll be around for weeks, years, et cetera. Another basic platform we use are etched silicon fibers, and I'll show you some data on those as well. The carbon fibers are from a bottom-up growth technique where we deterministically grow and shape these fibers. Here we use a top-down etching technique to create these. Now, if we could put one down, we could certainly shape them into other structures. Here we've got carbon nanofibers, and we outline basically our container sizes. Down on the bottom, you see the etched silicon structures. And so we could prepare isolated cells or arrays of cells. And this is now the physical environment we want to create for inserting the DNA molecules in and recreating biological networks. So the next challenge is, of course, how do you fill these kinds of structures and seal them? This is one of the fabrication approaches using the carbon nanofibers. And basically what it involves is just a combination of conventional MEMS processing, photolithographies, et cetera. In this case, we define catalyst paths from which the carbon fibers grow. We typically grow these a little taller than the structure with the carbon fibers. And then what we'll do is we'll use inkjet-based technologies or micromanipulators to put very small droplets inside individual wells. In this case, we have an array. This checkerboard is actually an array of carbon fibers, and this is the channel. We'll spit little droplets in there, picoliter droplets dry like that, so another challenge we had to overcome. In this case, we've got latex beads, nanometer-sized beads in each compartment. We seal this with a layer of PDMS. It's a soft polymer. Then we could wet the fluidic channels, and what you'll see is that the contents stay contained within their individual cellular environments, but are in fluidic communication with their neighbors. So the volumes in this case are on the order of about 250 picoliters. That's about a 10 micron tall cell height here with a 250 micron square on edge. Oops. Now, most of our efforts over the years have been really defining and characterizing the membrane properties. And there are numerous papers on a lot of these different subjects, and I won't go through them. But basically what we want to do is start defining membrane properties by controlling chemical coatings. We've developed techniques for coating these with biomolecules and polymers, et cetera. And also to study, say, electrical properties. There's phenomena it's called Debye screening, is this charge layer gets larger than the pore size, you get ion-perm selectivity. And so we developed devices to help us figure out how that works and how to control that. Now, physical size has been one of the key challenges. How do you make these, even though they might look impressive on the 5 micron scale, how do you bring them together in the order of tens of nanometers? So a few ways to do that. You could use various expensive lithography approaches, such as electron beam lithography. You can control the growth process of the carbon fibers. We pattern subsequently with oxide coatings. We use combinations of all the above. And then, of course, the next challenge is how do you characterize them? And we do some of that with flow measurements. We do some of that with advanced laser imaging techniques, laser-based imaging techniques. And I'll show you just a couple slides on that. In this case, this is an etched silicon membrane. You're looking at it from the top view. From electron imaging, the name just escaped me, but basically we collect these images and we could measure that, of course, and we're getting these down to about 15 nanometers in slit widths. Now, it's pretty tough to take your electron micrographs, is what I'm trying to say, and know what you're looking at. There's some taper to these edges as well. And so what we'd like to do is experimentally characterize the transport. And what we do there is we'll go ahead and dispense dye or fluorescein or whatever you'd like, green fluorescent protein. And we do a very common laser technique for cell biology. We do FRAP. And basically what that involves is you laser bleach what's inside the cell, and then you monitor the fluorescence recovery. And from that, you can see as we change the pore sizes on these, we change the recovery curves. And what we do is we couple that with simulations, and it allows us to extract out things like anomalous diffusion coefficients, as well as what the real transport is. And so anomalous diffusion is actually something we see on these scales. Anomalous diffusion is where there's a time dependence to your diffusion coefficient. And so your diffusion coefficient actually changes as a function of time and also space. And so it helps us to interpret the diffusion rates as well as engineer the flux of these containers. So obviously we could change the number of slits or the number of pores as well as the volume of the structure. This is an example of some recent work that's just been published. We'd like to add interfaces to these systems such that we could interact with them in real time. I'm sure to this community, microelectronics is old school, but I think it's going to be around for a little while. And what we've been trying to interface are electrical control mechanisms to our membranes. And so what we have set up here is just simple electrical pads with counter electrodes. And what we do is we grow the fibers, and since we can electrically address them, we could polymerize, electropolymerize coatings onto them. In this case, it's polypyrrole. And so we could actually define the coatings very precisely. And what's neat about polypyrrole is by application of a small voltage, we can cause that coating to shrink or expand. And so what you're looking at here is just the flow of 50 nanometer beads across a fluidic structure, and this black square is a carbon nanofiber-based membrane structure that's been coated with polypyrrole. And so the background glow here is the fluorescence of the beads. And then upon application of a voltage, we close up the membrane pores, and you can see how the beads basically build up against the wall here and are excluded from the container. That's because there's still flow going through the structure, but the pore sizes now have been reduced below 50 nanometers, and so the majority of the beads get collected inside the container. Now, one of the issues with the carbon nanofiber-based platforms is that since it's a deterministic growth process, you get a little more error into the placement of the fibers, as well as some of their characteristics. There's tilt involved, et cetera. And so we've started to look more and more at the cryo-etching techniques to etch E-beam defined structures. And so in this case, you're looking at silicon post structures that were defined by cryo-etching, or E-beam lithography followed by cryo-etching. And again, we've done the same tricks, a little different processing. We coat these with gold. We extend the pads out. And this is a top view of those fibers, and it gives you an idea of the scale at which we're at here. So we could take these and shrink them down such that the interfiber spacing now is on the order of about 80 nanometers after being coated with polypyrrole. And again, you could take the 50 nanometer beads and flow them through, and you can see the little black plume from when that closes. What's also been neat is we've taken this down to the scale of capturing proteins now. So in this case, we've flowed fluorescein-labeled bovine serum albumin through there, and that's what this green is. It's now captured within the device and held there. We'll hold it there for whatever length of time. Then open the structure, and what will happen is the material then leaves the device. And so this is essentially where we're at in designing some of the membrane structures and adding active components to their opening and closing. So another effort in the group, of course, is getting these reaction systems to work inside containers. And here again, we've taken some simple steps. This is just a picture of a very simple fluidic device. It's a single channel running through, and we have inlets and outlets, and it's a few centimeters across. Inside that channel is an array, a linear array of these individual containers. And what we do here is we place enzymes inside it, in this case horseradish peroxidase, and we're just simply running Amplex Red, which is a fluorogenic reagent for detecting this reaction, along with some peroxide. And what you'll see in the structures is that the enzymes stay within the structures for the most part. The reactions occur and the devices glow. And then some of that Amplex Red will leak back out. So you can imagine a variety of applications, but again, we're heading towards how to create systems. Now, if we could contain Proteins, we can contain DNA. In this case, it's just an optical micrograph, fluorescent micrograph of different concentrations of a serially diluted plasmid that we then subsequently stain with lithium bromide. The DNA molecules will stay in there for quite a long time under flow. They, for the most part, don't leak out. They're significantly larger, and of course, they're a different structure than globular proteins. But with this ability to contain DNA, we can now start looking at protein expression in these devices. And this is certainly a work in progress, but in this case, we'll take our DNA construct and it'll be expressing the gene for green fluorescent protein, and we'll put this under a T7 promoter. And we'll take devices and we'll put DNA in some and not in others. And we'll add the cell-free extract to the devices and look for the expression of DNA, and that does work. There's a leakage problem with a lot of these. The RNA tends to leak out, and a lot of times you get expression out here in the channel as well. But like I said, this is how we learn how to make it work. We've also done a lot of these in sealed containers. In this case, there's no pores to the chamber. It's just simply a chamber. And here again, we'll start looking at how to control DNA templates in there and what their effect is on protein expression, as well as, of course, the time-dependent expression of these proteins. And so we look at this as some of the first steps for assembly and testing of genetic networks. And so I think I'm keeping to my 20 minutes, but just one more slide here, and this is just, again, where we're at with this sort of nano-enabled synthetic biology. Basically, I mean, we're excited because nanotech has really given us a lot of these very enabling tools for understanding and emulating biological function and design. Interfacing the molecular and cellular level, mimicking cell structures, et cetera. And of course, it's starting to look at hypotheses about how these systems are created and organized. And I think ultimately it's going to lead to the development of these devices to help us understand system design. And biomedical sensing and actuation is certainly what we pitched to the NIH when we got this funded. But it's also getting in towards how do you do biochemical transformations as related to bioenergy concerns, or perhaps synthesis of specialty chemicals through metabolic processes. So with that, let me just acknowledge my colleagues. It's a fairly good-sized group. We do various things. These are most of the people involved in the cell mimic work, and the support for this has been mostly from the NIBIB with early support from DARPA, as well as, of course, the Department of Energy and the Center for Nanophase Material Sciences. So I thank you for your attention.